Good morning. Today we will be presenting a very recent discovery made about the ability of Staphylococcus aureus, a potentially harmful microbe of the skin, to undergo adaptive evolution in eczema patients going through treatment. So what is eczema? Eczema, or atopic dermatitis, AD for short, is a common chronic skin inflammation that causes the skin to become dry, itchy, and bumpy due to a defective skin barrier and an altered immune response. This results in skin that's more sensitive and vulnerable to irritants, allergens, and environmental factors. In Singapore, it affects about 1 in 10 adults and 1 in 5 children. Our skin microbiome is home to millions of microbes, one of which is Staphylococcus aureus. There has been substantial evidence showing that abundance of S. aureus on the skin is proportional to the severity of AD. It is estimated that between 30 and 60% of people carry S. aureus in their nostrils, where it is usually harmless, but in people with eczema, S. aureus often spreads to eczema patches and infects the skin, which worsens disease severity by promoting skin damage. This led Associate Professor Dr. Tammy Lieberman from the Massachusetts Institute of Technology to ask, does S. aureus need to change to live on skin affected by eczema? Can we learn something about how these bacteria are interacting with atopic dermatitis skin from watching its evolution? These questions formed the basis of a research team study as they sought to find mutations which help S. aureus adapt to living on the skin of AD patients. This could robustly identify the individual mutations emerging in disease states and help us better understand the potential impact of de novo microbiome mutations on complex diseases.
Moving on to the study design, it comprises of sample collection, whole genome sequencing, and analysis. The study focused on 23 children aged 5 to 15 years old in Mexico with moderate to severe eczema. Skin swabs were collected from children over the course of 9 months. Samples of the skin microbiota were taken from 7 skin sites, 4 from skin sites affected by eczema, and 3 from unaffected skin sites. Over the 9 months, children were also given standard treatments for eczema, such as topical steroids and bleach baths. From each sample, genomes of S. aureus cells were sequenced. For analysis, researchers first determined the minimal number of independent colonization of S. aureus onto each child. To do so, S. aureus cells were grouped based on their genetic similarities into lineages. Cells separated by less than 100 mutations across the core genome, which includes conserved essential genes, are grouped together. Each lineage comprises of cells that theoretically arose from a single colonization of S. aureus. For each patient, phylogenetic trees were constructed to illustrate the accumulation of mutation inside each lineage. The tree shows that the evolution of S. aureus from a hypothetical initial colony that colonized the skin. To determine whether mutations confer a survival advantage to S. aureus on AD skin, researchers identified signatures of on-person adaptive evolution. Adaptive signatures include mutations that arose independently in the same gene across separate S. aureus cells within a patient. Lastly, researchers sought to determine if the adaptive mutations identified provide an advantage for S. aureus specifically on AD skin. They analyzed publicly available whole genome of S. aureus from the skin of AD patients, healthy carriers, and people who had acute S. aureus infections.
Let's move on to the results. Here are the results from the researchers' longitudinal tracking of S. aureus lineages over 9 months. Researchers found that the skin of most patients was colonized by one stable S. aureus lineage, which is illustrated in this figure. 6 out of 23 patients provided a high quantity of isolates, which were subsequently used for more in-depth analysis of mutation accumulation. Researchers calculated the time since major lineages isolated shared the same ancestor, TMCRA, as seen in this figure. Results showed that most patients had low TMCRA values, suggesting that current lineages were found were new. The study also showed that mutations resulted in within-lineage change in genotype as opposed to divergence into coexisting lineages. Further visualization using Muller diagrams shown here allows us to see that newly acquired mutations replace existing varieties. Researchers had concerns that the new source of mutations in lineages could have been due to transmission from an external source as opposed to on-person mutation. However, the construction of phylogenies showed mutation accumulations in small stepwise successions, providing evidence that this was not the case.
Given the mutations observed across the S. aureus lineages, the researchers hypothesized they might confer a competitive advantage on AD skin. From analyzing the signature characteristics of the different mutated genes, they found that patient 12 had 4 different mutations in the CAPD gene, marked by the black stars in this figure. This high mutational density is significant considering the overall number of mutations in this patient, thus suggesting that CAPD mutations might have an adaptive role on AD skin. One of the CAPD mutations identified, termed E199, introduces a premature stop codon, giving a truncated polypeptide. This E199 mutation increased in frequency over time, shown by the increasing thickness of the polygon, which eventually replaced existing diversity. Similarly, another CAPD mutation called N595S, observed in patient 9, also exhibited a similar trend, as seen here. No comparably strong signals of de novo adaptive mutations in other S. aureus genes were detected across the patient cohort. Collectively, the CAPD mutations observed within patients, across patients, and how they spread and replace existing diversity thus suggests that alterations in CAPD provide a survival advantage for S. aureus on AD skin. So what exactly is CAPD? The CAPD gene codes for an enzyme involved in the first step of capsular polysaccharide synthesis in S. aureus, where this sugar coat helps shield the pathogen from detection by immune cells. Truncation mutations in CAPD thus result in capsule loss. Apart from the 2 patients with de novo adaptive sweeping CAPD mutations demonstrated previously, further analysis showed 4 other patients who had ancestral truncation mutations in this gene, meaning the capsule loss mutations had already arisen prior to this study. 4 others also showed ancestral capsule loss driven by other types of mutations. Meta-analysis performed across 4 other literature studies showed that percentage of CAPD truncation was significantly increased in AD patients compared to healthy controls. Several independent occurrences of CAPD truncations in AD patients, boxed in pink, were also observed. Taken together, these results support the previous findings, suggesting that capsule loss is favored on AD skin, and these mutations in the capsule can drive S. aureus adaptation in AD patients.
Moving on to the discussion. The observed findings are interesting considering that there is no consistent association between S. aureus sequence types, clonal complexes, or other such groupings with AD, but yet there is apparently selection pressures exerted on the bacteria of AD sufferers. For instance, there is the apparent selection for capsule loss, but acapsular groups do not dominate in the colonization of patients. In this case, it is suggested that there may be selective trade-offs to generating or not generating capsule in AD, with a given phenotype selected for depending on the context. The effect of person-specific selection should also not be ignored. Importantly, due to the relatively small size of the cohort, as well as other factors contributing to AD severity, this study was unable to directly link S. aureus encapsulation to disease severity. A larger sample size would be better suited to assess this. Taking into account the complex selection pressures mentioned earlier, a larger cohort would also help uncover additional variations and adaptations associated with AD beyond capsule status. Furthermore, as all the patients underwent treatment, it is uncertain to what extent this contributed to the bacteria's evolutionary path. However, the author do note that mutations in CAPS-D were observed in patients who both did and did not receive bleach baths as treatment. Regarding the translational potential of this work, small molecules could be screened and used to target unencapsulated S. aureus in order to reduce colonization in AD. However, this would require a better understanding of the association between unencapsulated bacteria and disease severity. It is worth noting that this treatment may have unintended consequences, such as selecting for encapsulated strains that are more virulent in other contexts. Nevertheless, there is value in this approach, as the individual mutations selected for in disease states could become potential therapeutic targets. And that brings us to the end of this Journal Club presentation. Listed here are the main findings of the article. And here we have our references. Thank you very much for listening.